By the early 1950s, the question of DNA's structure had become one of the most urgent problems in biology. Scientists already knew that DNA carried genetic information, but without understanding its physical form, they could not explain how this information was stored, copied, or passed on. Solving this mystery required the combined efforts of chemistry, physics, and biology, as well as new experimental tools that could reveal structures too small to be seen under a microscope. One of the most powerful techniques available at the time was X-ray diffraction. This method allowed scientists to infer the three-dimensional structure of molecules by analyzing how X-rays scattered when they passed through crystallized samples. At King's College London, Rosalind Franklin became a leading expert in this technique. Her careful and precise work produced exceptionally clear diffraction images of DNA fibers. These images revealed patterns that strongly suggested a helical structure. Franklin discovered that DNA existed in two forms, depending on its water content. One form, later called aDNA, appeared more compact, while the other, known as bDNA, showed a distinct repeating pattern. Her most famous image, often referred to as photograph 51, provided critical information about the symmetry and dimensions of the DNA molecule. From this image, it was possible to calculate the spacing between repeating units and infer the presence of a helical structure. At the same time, at the University of Cambridge, James Watson and Francis Crick were attempting to solve the structure of DNA using model building rather than direct experimentation. They believed that constructing physical models based on known chemical constraints could reveal how DNA was organized. Their approach relied heavily on data produced by others, including chemical measurements and X-ray diffraction results. Although Franklin had not intended her unpublished data to be widely shared, some of her findings were shown to Watson and Crick without her direct knowledge. These data, combined with other experimental evidence, proved crucial. They confirmed key features such as the helical nature of DNA and the regular spacing of its components. This information helped Watson and Crick refine their model and avoid earlier mistakes. In 1953, Watson and Crick proposed the famous double-helix model of DNA. According to their model, DNA consists of two long strands twisted around each other in a spiral shape. Each strand is made of repeating units called nucleotides, composed of a sugar, a phosphate group, and a nitrogenous base. The sugar and phosphate form the backbone of the molecule, while the bases face inward. A crucial insight of the model was the specific pairing between the bases. Adenine always pairs with thymine, and guanine always pairs with cytosine. These base pairs are held together by hydrogen bonds, creating a stable yet flexible structure. This complementary pairing immediately suggested a mechanism for replication. If the two strands separate, each can serve as a template for creating a new complementary strand. This idea elegantly explained how genetic information could be copied with high accuracy during cell division. It also clarified how information could be encoded in the sequence of bases along the DNA strand. The order of these bases formed a kind of biological language, capable of specifying the structure and function of proteins. With this realization, the mystery of heredity began to make sense at a molecular level. Watson and Crick published their model in the journal Nature in April 1953. Their paper was remarkably short but profoundly influential. In it, they famously noted that the specific pairing of bases immediately suggested a copying mechanism for genetic material. This understated sentence hinted at a revolution in biology that would soon unfold. The contribution of Rosalind Franklin, however, was not fully recognized at the time. She continued her research on virus structures and made significant contributions before her death in 1958. Because the Nobel Prize is not awarded posthumously, she was not included when Watson, Crick, and Maurice Wilkins received the Nobel Prize in Physiology or Medicine in 1962. Today, her role is widely acknowledged as essential to the discovery of DNA's structure. The unveiling of the double helix marked a turning point not only in genetics but in all of biology. It provided a unifying framework for understanding how information flows within living systems. The structure explained mutation, replication, and inheritance in a single, coherent model. It also laid the groundwork for future discoveries in molecular biology, including the genetic code and the mechanisms of protein synthesis. From this point onward, biology entered a new era. Researchers could now investigate life at the molecular level, tracing the flow of information from DNA to RNA to protein. This conceptual framework, often called the central dogma of molecular biology, became one of the foundational principles of the life sciences. The journey from Miescher's discovery of nuclein in 1869 to the revelation of the double helix in 1953 illustrates how scientific understanding evolves over time. It is a story shaped by curiosity, persistence, collaboration, and occasional controversy. Each generation of scientists built upon the work of those before them, slowly uncovering the secrets hidden within the cell. In the final part of this series, the focus will shift to the impact of DNA discovery on modern science and society, including medicine, technology, and our understanding of what it means to be human. After the discovery of the double helix structure in 1953, the study of DNA entered a new and transformative era. What had once been an obscure substance found in cell nuclei became the central focus of modern biology. Scientists now possessed a model that explained how genetic information could be stored, copied, and transmitted. From this foundation, an explosion of discoveries followed, reshaping medicine, technology, and humanity's understanding of life itself. One of the first major breakthroughs after the double helix was the deciphering of the genetic code. Researchers sought to understand how sequences of DNA bases could specify the production of proteins. During the late 1950s and early 1960s, scientists such as Marshall Nirenberg and Hargoband Khorana demonstrated that groups of 3 DNA bases, known as codons, correspond to specific amino acids. This discovery revealed how information written in the language of DNA is translated into the proteins that perform nearly all functions in living cells. This understanding led to the formulation of the central dogma of molecular biology, which describes the flow of genetic information from DNA to RNA and then to protein. DNA serves as the long-term storage of information, RNA acts as a messenger and intermediary, and proteins carry out structural and functional roles. This simple yet powerful framework unified many previously disconnected biological observations and provided a roadmap for future research. As molecular biology advanced, scientists learned how DNA replicates, repairs itself, and mutates. They discovered enzymes responsible for copying DNA with remarkable accuracy, as well as mechanisms that correct errors when they occur. At the same time, researchers began to understand that mutations, though often harmful, are also the source of genetic variation and evolution. Changes in DNA sequences, passed down through generations, help explain the diversity of life on Earth. The practical applications of DNA research soon began to emerge. In medicine, understanding genetic mechanisms opened the door to diagnosing inherited diseases. Conditions such as sickle cell anemia and cystic fibrosis were linked to specific genetic mutations. This knowledge transformed medical genetics, allowing doctors to identify carriers, predict disease risks, and improve patient care. Over time, genetic testing became a powerful tool for early diagnosis and prevention. DNA research also revolutionized forensic science. In the 1980s, techniques for analyzing DNA profiles made it possible to identify individuals with extraordinary accuracy. DNA fingerprinting began to be used in criminal investigations, helping to solve crimes, exonerate the innocent, and identify victims. What once began as a laboratory discovery became a cornerstone of modern justice systems around the world. In agriculture, knowledge of DNA enabled the development of genetically modified organisms. Scientists learned how to insert specific genes into plants to improve resistance to pests, increase nutritional value, or enhance tolerance to environmental stress. While controversial, these technologies have had significant impacts on global food production and continue to influence debates about sustainability and ethics. The rise of biotechnology in the late 20th century further expanded the influence of DNA science. Techniques such as recombinant DNA technology allowed genes to be cut, copied, and inserted into other organisms. This made it possible to produce human insulin in bacteria, revolutionizing the treatment of diabetes. Similar methods led to the development of vaccines, hormones, and therapeutic proteins that have saved millions of lives. Perhaps one of the most ambitious DNA-related projects was the Human Genome Project, launched in 1990. Its goal was to determine the complete sequence of human DNA, containing approximately 3 billion base pairs. Completed in 2003, the project provided a reference map of the human genome and marked a milestone in scientific history. It revealed that humans share the vast majority of the genetic material and that relatively small differences contribute to individual uniqueness. In recent years, advances such as CRISPR-Cas9 gene editing technology have further transformed the field. Scientists can now edit DNA with unprecedented precision, raising possibilities for curing genetic diseases, engineering cells for therapy, and reshaping biotechnology. At the same time, these powerful tools have sparked ethical debates about the limits of human intervention in nature. Looking back, the journey that began in 1869 with Friedrich Miescher's discovery of nuclein appears extraordinary in its scope and consequences. What started as a careful chemical observation in a small laboratory grew into a global scientific revolution. Each generation of researchers added a new layer of understanding, transforming DNA from an unknown substance into the blueprint of life. The history of DNA is not only a story of scientific progress but also a story of patience, curiosity, and collaboration. It reminds us that even the most groundbreaking discoveries often begin quietly, without immediate recognition. Miescher could not have imagined that his work would one day influence medicine, law, agriculture, and technology on a global scale. Today, DNA stands at the center of modern biology, connecting the past, present, and future of life on Earth. From evolution to personalized medicine, from ancient ancestry to future gene therapies, the legacy of the 1869 discovery continues to shape human knowledge. The story of DNA shows how a single discovery, pursued with dedication and curiosity, can change the way humanity understands itself. And this is the story of a discovery that began quietly in 1869. With a substance no one truly understood. From a mysterious material hidden inside the nucleus, to the very code that defines every living being on Earth, DNA was not discovered in a single moment of glory. It emerged through decades of patience, doubt, failure, and perseverance. Through the work of scientists who asked questions long before answers existed. From Miescher's first isolation of nuclein, to the experiments that proved DNA carried heredity, and finally to the revelation of the double helix, each step reshaped our understanding of life itself. Today, DNA is more than a molecule. It is the foundation of modern medicine, the key to solving crimes, the map of evolution, and a guide to the future of humanity. This story reminds us that the greatest discoveries often begin in silence. With curiosity, patience, and the courage to explore the unknown. If you enjoy deep historical stories, science documentaries, and the ideas that changed the world, don't forget to like, subscribe, and join us on this journey through time. Until the next story, keep questioning, keep learning, and keep exploring.